everybody, welcome to my YouTube channel. Today I am going to provide you a notes that will be quick new revision notes for the class 12th chapter 2, that is reproduction in flowering plant. Okay, so first we will start with about flower. Okay, flower is an organ which help in sexual reproduction. Two whorls are present, which is further divided into two parts, inner and outer. Outer whorl non-essential whorls. Two part of flower comes under this, that is calyx and corolla. They are non-essential because they do not take part in sexual reproduction. Second, it is inner whorl, that is essential whorls. Two parts come under this also, that is symposium and gynosium. They are essential because they take part in sexual reproduction. Now next is the structure of flower, which you should draw and which you should remember for board exams. Okay, now the most important point related to competitive part, that is when sepals and petals are not differentiated, that means that they remain undifferentiated, whorls are known as perianth and each number of perianth is called sepal. Now next slide. There are two phases which generally occurs, that is pre-fertilization and post-fertilization. In pre-fertilization, three steps are there: stamen, microsporangium, polymorph. So first we will start with the stamen. It consists of two parts, filament and anther. So next, let's know something about the stamen. Stamen. It is a male reproductive unit of angiosperm. Each pollen sac contains number of pollen grains. The four pollen sac lie in four corners of a dithecous anther. Here might be there something doubt related to dithecous anther, that is cleared out in next line. Dithecous anther and anther lobe joined together by a non-sporangious tissue called connective. Next point, sporangious tissue means spore producing tissue and non-sporangious is just reverse, means that it is not a uh, uh, spore producing tissue. Now, anther development. The anther development starts from a mass of homogeneous meristematic cells surrounded by a epidermis. Second, four lobes are formed and four layers of archisporal cells are differentiated. Archisporal cells means a cell or group of primitive cells that form cells from which spores are developed. Each archisporal cell divides to form two types of cells. So two types of cells are from archisporal cell. First is primary parietal cell and the second one is primary sporangious cells. Okay. Now fifth point, each parietal cells divides several times to form anther wall and sporangious tissue forms the microspore or pollen mother cell. Now it will be cleared. Second, the walls of anther. First wall is epidermis, that is a single layer of cells and protective in function. Second, that is epidermis. Second wall, that it is also of single layer. It helps in anther descent. Anther descent means release of pollen grains. And third, that is middle layer. It ranges from 1 to 6, means 1 to 6 layers are of middle layer. It degenerates at the time of maturity of anther. Means its main function it is that to degenerate at the time of maturity. Now the fourth one is tapetum. That is the most important layer for the board exam because several times the questions have asked from tapetum. Its structure is pyramidal and it surrounds the sporogenous tissue. Here is diagram related to this, which you have to draw in your copy. Once again I am showing you the diagram so that you can draw it or you can take a screenshot also. Okay, so this is the diagram. Now we should move forward. Now the role of tapetum, I put stars only in those points which are most important for the board exam. Okay, so first, nutrition. It provides nourishment to the developing microspore. Second point, it secretes enzyme callose that dissolve callose thickening to release microspore from microspore tetrad. That means it helps to release its microspore from microspore tetrad after dissolving the thickening of Callose enzyme. Third point, it produces ubiquitous body that helps in development of pollen grain present in cytoplasmic part of tapetum. Or in simple word, you can say that it helps in the it helps in pollen development, and that is coated with sporopollenin. Sporopollenin is a hard cover and forms exine of pollen grain. Exine is one of the layer of pollen grain which helps for the formation of pollen grain. Fourth point, formation of pollen tube in certain plants like China rose. Means tapetum layer helps in certain plants for the formation of pollen tube. Now the types of tapetum. There are two types, glandular tapetum and amoeboid tapetum. Glandular tapetum found in angiosperm, secretory in nature, and amoeboid tapetum common in lower monocot and dicots and is from Plasmodium. And the amoeboid tapetum's another name is Plasmodial tapetum. Okay. Note one point important which is very important for you all, that is endothecium is a second layer of anther having callose thickening which holds together the microspore. As you have learned earlier only, that it helps to release the microspore from the microspore tetrad which helps to hold it together. Okay, so this was in the pre-EF, it's in first part. Second part I will cover out all the other remaining topics. Thank you for watching my video. Like, share and subscribe. And if you have any suggestion, please comment in the comment box. And other remaining parts will be covered in second part of the video. Okay. Thank you.